Hello dear learners, today we are going to talk about beta-lactam antibiotics. Beta-lactam antibiotics, as you all very well aware about this term beta-lactam. So we are going to cover in today's video about beta-lactams, structure, their classification, their mechanism of action, their mechanism of bacterial resistance, uses and adverse effects. So in this particular video, to start with, first we will understand why beta-lactam antibiotics are known as beta-lactam antibiotics, because they contain a ring which is known as beta-lactam ring. Like if you will see this ring, 4-membered ring which they have highlighted and this they have enlarged, sorry. So this is beta-lactam ring, which is a 4-membered ring. So this beta-lactam ring is responsible for the name beta-lactam antibiotics, and nitrogen is attached to the beta carb relative to the carbonyl ring, and hence the name. Since the nitrogen which is there, which is attached at the beta carbon and which is relative to the carbonyl ring, and hence the name is beta-lactam ring. So this is beta-lactam ring. So beta-lactam antibiotics, if you will see, basically they have two major groups. First group is penicillin, second group is cephalosporin. In case of penicillin, if you will see, this is the beta-lactam ring, as well as in cephalosporin, this is the beta-lactam ring which we have seen just now. So if you look at the parts of the structure, now this is beta-lactam ring, this is thiazolidine, thia and aza, this is thia and aza, thiazolidine, and this, since thiazolidine, it is beta-lactam ring, and this is acyl side chain, and this is C double bond ONH. So here you can see this will be common, like from here till this, this is the free carbonyl group COOH, this COOH which you can observe, and at 6th position. Now how the position is 6th now? If you will start the numbering, nitrogen 1, 2, 3, 4, 5, then 6 and 7. And in case of cephalosporin, numbering again from nitrogen, but since it is 6-membered ring, here it is 6 and then 7th position will come to the beta-lactam ring. So this 6th position contains this RCONH. This R is R can be varied, CONH is constant, here also CONH is constant, and R can be varied in case of cephalosporins. And this free carboxyl group is the basic structure. This was with respect to penicillin and cephalosporin. Penicillins, they contain thiazolidine, which is 5-membered ring, and cephalosporin, they contain 6-membered ring, which is nothing but dihydrothiazine ring. Thia-aza. So dihydrothiazine ring. That means there is a presence of saturation where would have been the double bond present here in this position. So these are the two major groups. Other beta-lactam antibiotics are the classes like penams, cephems. Now penams, as the name suggests, penams, penicillin. So it is benzyl penicillin, phenoxymethyl penicillin, that is penicillin G, penicillin V, cloxacillin, amoxicillin. And in case of cephems, if you will see, it is cephalothin, thin banna hai, lexiflexi banna hai, whatever the trick we have covered, and cephalosporin C. So it is cephems, penams and cephems, penicillins and cephalosporins. Next beta-lactams are carbapenems and penams. Now here it is carbapenems and penams, biapenam, doripenam, tebipenam. Then monobactam class is there. From monobactam class it is tigemonam, azithrionam, etc. Then beta-lactamase inhibitors such as clavulanic acid, tazobactam, sulbactam, these are the enzyme inhibitors, beta-lactamase. So if you will compare the structures of these like penicillin, cephalosporins, monobactam, carbapenems, so you can see here in the structure, see beta-lactam ring, this one is beta-lactam ring, this is beta-lactam ring, this is beta-lactam ring. So it will remain common in all the beta-lactam antibiotics, this ring, beta-lactam ring. Now these comparative which we have seen, penicillin, cephalosporin, carbapenems and monobactams. So penicillin, cephalosporin, we just now discussed earlier that penicillin contains 5-membered heterocycle and cephalosporin contains 6-membered heterocycle. Again carbapenem, if you will see, it is 5-membered, and monobactam, it is only SO3H. Rest all if you will see, the left hand side is same, beta-lactam and CONHR. And here is the description, beta-lactam ring which is joined to a 5-membered thiazolidine ring, that is penicillin, 6-membered dihydrothiazine ring, that is cephalosporin. In carbapenems, like here in this example which you can see, it is carbapenems, the beta-lactam ring is also joined to a 5-membered ring, although it is carbocyclic. Here 5-membered ring, but it is carbocyclic. Then monobactams have a monocyclic beta-lactam structure. This monobactams have monocyclic beta-lactam structure, like only beta-lactam and the side chain, and here it is SO3H sulfonic acid. So these are beta-lactam ring which are exhibiting the antibacterial properties. Now the suffixes, if you will see, they end with, like other antibiotic classes are there, aminoglycoside, tetracyclines, macrolide, fluoroquinolone, cephalosporins. So aminoglycosides ends with mycins like erythromycin, azithromycin. Then tetracycline ends with cyclines. Then macrolides ends with thromycin. Then fluoroquinolone ends with floxacin, ofloxacin, clofloxacin. Cephalosporin starts with ceph, everything. So these are few of the easy ideas to remember the suffixes, whereas in beta-lactam you will not find such suffixes because the classes are like penicillins, again subclassification is there, cephalosporin, again subclassification is there, like this classes are there. Now moving towards the ring formation, here in case of beta-lactam ring formation, how this ring has been formed, that we will see now. Since as you can see here, beta-lactam, one blue part is there and one pink part is there, or red part you can call. So this is beta-lactam ring, and this is the fusion of cysteine and valine, as you can see here. Now cysteine structure you can find out, NH2, then CH2SH and free carboxyl. Here also NH2, then CH2, substituent will change, CH3 and carboxylic acid as a free group. So this part, if you will properly observe, this has clubbed over here. Then this part, SH wala part, has been clubbed over here. So it is cysteine plus valine forming the beta-lactam ring. Now if you will come towards the penicillinic acid, 6-amino penicillinic acid, which is there in the penicillins. Now penicillins, 6-amino penicillinic acid is there, like at 6th position they have the acyl side chain, and this R will vary from penicillin to penicillin, which we have discussed in detail in our earlier penicillin video. Like if it is CH2 benzene ring, it is benzyl penicillin. Then if it is CH2O phenyl ring, then it is phenoxymethyl. So it is penicillin V. So this way the members are, like here if you can see penicillin G and V, G contains CH2 phenyl ring and penicillin V contains phenoxymethyl penicillin. This is phenoxy and this is methyl. Other members which we have discussed in penicillin video, other semisynthetic members are ampicillin, amoxicillin, carbapenems, oxacillin, cloxacillin and dicloxacillin. Only substituents will vary. Now this substitution we have already gone through in the penicillin case. So I will not much focus on these structures, and same it is there in the cephalosporins. As in case penicillins, it is 6-amino-penicillinic acid. In case of cephalosporins, it is 7-amino-cephalosporanic acid, because here comes the 7th position, not a 6th position like in penicillin. Why? Because this contains 6-membered heterocycle. So it is 7-amino-cephalosporanic acid, and these are the substituents which can be present on the structure. So basic nucleus of cephalosporin is 7-ACA and basic nucleus of penicillin is 6-amino-penicillinic acid, that is 6-APA. Here it is 7-ACA. Now coming towards the various cephalosporin substituents, if you will find, this is the general structure, which is nothing but a 6-membered ring and beta-lactam ring. Now this is this R1 and R2, there are 2 substituents. So if you will find the substituents, I will take only one example which I have highlighted, cephadroxyl. Here you can see cephadroxyl. R2 is CH3 and R1 is phenyl. This is benzene and this is hydroxy, and here it is, since it is attached to this CH2, it is OH ring, and here it is CHNH2, CHNH2 and CONH will continue in the structure. So these are the substituents, and this is called as cephadroxyl. Like this, various substituents are there: cephachlor, cephalexin, ceftrazine, then cephazidone, cefapyrine, etc. Now coming towards the mechanism of action. Now this mechanism of action, which is the common mechanism of action, we have already covered in the earlier video, like mechanism of action of antibiotics based on the mechanism of action. This diagram you can find, and there are various subclasses like cell wall synthesis inhibition, then cell membrane damage, then folate synthesis, that is metabolic inhibitor, protein synthesis inhibition, wherein it plays a role in DNA, RNA and with ribosomes. So these are various classes. If you will see, or if you will recall all of them, like first is cell wall synthesis, since it is the outermost membrane. If there is a damage to the cell wall, then cell wall will not remain, then the survival is critical of the bacteria. So it is inhibition of cell wall synthesis, and the antibiotics which are working by this way are beta-lactam, like penicillin, cephalosporins, bacitracin and vancomycin are also there. Then inhibitors of cell membrane, second layer is cell membrane. As we already know, cell membranes are also one of the important barrier. So entry and exit is regulated, that is transport is regulated by the cell membrane, which is permeable. So it will allow only selective substance to enter inside and toxins to leave outside. So the antibiotics which are acting by this way are polymyxin B and colistin. Then protein synthesis. In case of protein synthesis, if you will see, ribosomes, DNA, RNA and actual protein synthesis, the 30S and 50S subunits working. So it is inhibition of protein synthesis. Since all of you know that protein synthesis is a very essential process which is must for the multiplication of the bacteria and survival of the bacteria. So it can be inhibited by ribosomal binding, like binding with the 30S and 50S subunit of the ribosome. The activity can disrupt the normal metabolism, as well as growth and multiplication also will be inhibited due to the protein synthesis inhibition. Examples are aminoglycosides, macrolides, lincosamides, streptogramins, chloramphenicol, tetracycline. And next, if you will see nucleic acids like DNA and RNA. So here it is, DNA and RNA also plays a role in the replication. So there are some antibiotics which works by binding with DNA and RNA synthesis. So interference in the normal cellular process will compromise the bacterial multiplication and survival. The examples are rifampicin or rifampin and quinolones or metronidazole, etc. So this is cell wall, this is cell membrane. If you will see, this is metabolic pathway, PABA to DHFA to THFA with the help of sulfonamides, like sulfonamides inhibiting these steps. So because of which metabolism is inhibited, then protein synthesis inhibition, then RNA synthesis inhibition, DNA gyrase inhibitors, then cell wall synthesis inhibitors. Metabolic processes, you are very well aware, which we have studied in sulfonamides. So it is inhibition of folic acid synthesis is the metabolic pathway by which inhibitors of metabolic processes are acting. Now coming towards the actual mechanism of action of that of beta-lactam antibiotics. Now beta-lactam antibiotics acts by disrupting the synthesis of bacterial cell wall and by interfering with the transpeptidase, that is plasma binding proteins, which catalyze the cross-linking process, and by interfering with the function of them like transpeptidase enzyme, this cell wall synthesis will also be inhibited. Along with that, it also increases the capacity of the autolysins or enzymes and its self lysis, you can call. Now as you can see here in the diagram, this is plasma binding protein transpeptidase subunit, this gray colored structure, and this is the cross linkage which is occurring between the cell wall like NAM, NAG. Okay, now NAM, NAG, NAM, NAG. So what beta-lactams will do? Beta-lactams will cross the membrane. Now how they will cross the membrane? We can see here, like see in case of gram-negative bacteria, in gram-negative they have a lipopolysaccharide layer and in which porine channels are embedded. So through porine channels they will enter and they will show their action. Whereas in case of gram-positive, where only peptidoglycan is there, lipid membrane is not there. So it is very easy, very simple to enter inside the cell. Then binds to the active site of plasma binding proteins and inhibit them. PBP को bind करके उनको inhibit करना is the major function. Now if you will see the chain, that is cross linking here. Now see, as you can see here, it is binding to the transpeptidase active site, and because of the binding, this cross linking is getting hampered, and because of which cell wall synthesis will not take place. Now here it is transpeptidase enzyme, that is plasma binding protein. If you will see, the cross linking is occurring due to the PBP, which is catalyzed by the transpeptidase. Now what will happen here? Here the beta-lactams will mimic the structure of d-alanyl-d-alanine link and bind to the active site of PBPs, that is plasma binding protein. That means beta-lactams will bind to the PBPs and they will disrupt the cross-linking process by interfering with the function of the PBP, that is plasma binding protein. Since beta-lactams and these are structurally similar. Next part of the mechanism is all the bacterial cell wall possess their associated enzyme called as autolysins. Autolysins, when they get activated, they will hydrolyze their own cell substances. So this capacity of autolysins to dissolve the peptidoglycan, it is essential for the cell growth, cell septation, sporulation, transformation, for these processes which is essential. So dissolving that is the major function of the autolysins, that is peptidoglycan. Now coming towards the uses, beta-lactam antibiotics, they have broad spectrum antibiotics also as well as narrow spectrum antibiotics also. So these are active against both gram-positive as well as gram-negative bacteria. Certain species of gram-positive such as Streptococcus, Staphylococcus infections, Clostridium, Corynebacterium species, and various broad spectrum antibiotics are also active against the gram-negative microorganisms like Neisseria gonorrhoeae, E. coli, Neisseria meningitis, Salmonella, Shigella, etc. So how you will remember this? Even if 3 also you remember 3 to 4, it is okay. Gram positive, the trick is SAC, and gram negative, the trick is James. James में it is G stands for gonorrhea, E stands for Escherichia coli, M stands for meningitis, and S stands for Salmonella, Shigella. So it is James. And SAC. SAC is S for Streptococcus, Staphylococcus, A for actinomycetes, and C for Clostridium, Corynebacterium. So initially S is also twice, C is also twice, and in between A, which is alone. So it is active against both gram positive as well as gram negative bacteria, and it helps in preventing and the treating bacterial infections caused by the susceptible organisms. Looking at the adverse effects, if you will see, this image is the self-explanatory image where you will find some GI intolerances such as nausea, okay, vomiting, then diarrhea is also there, okay. So these are gastrointestinal disturbances or intolerances. Then if you will see, most commonly rash, anaphylactic shock, like here you can call this rash, okay. Then renal abnormalities can also be there, renal failure can also be there. Then hemolytic abnormalities such as hemolytic anemia can also be there, neutropenia, eosinophilia, etc. Now these are common symptoms which are shown by the picture. The major of beta-lactam is rash and anaphylactic shock. With this we finish with the beta-lactam antibiotics introductory portion. Thank you.